Record reflects we are back in session. Parties and counsel are present. Mr. Attorney are present. Witnesses, staff on the witness stand. And you may proceed, Mr. McGee.
Thank you, Your Honor. Mrs. Ryan, you talked about different ways of processing evidence. What are the two ways that items can be processed for DNA?
So the way the items can be collected for DNA are going to be with what we would call traditional methods, which would typically be swabbing or cutting out a stain. And then another method that has been recently developed is something called the MVAC, and that's what this instrument here is.
I'm going to show up I just put up an image. The traditional method with cotton swabs.
Yes.
We had, I believe it was Ms. Williams who demonstrated for the jury how the tool items will work and you use the cotton swab. Can you just describe it again for the jury how that works basically?
Sure. So, I mean, there are different people collect different ways with a cotton swab. Some people just use one swab that's wetted with sterile water. One of the more common ways is something called a dual swab or a wet swab, dry swab method, where you would use one sterile cotton swab wetted with sterile water, and you would swab whatever item that you're interested in collecting DNA from. So that could be, the brim of a hat. It could be the collar of a shirt. It could be a door handle. Any item. It could be a gun, a knife. You're basically wetting that swab and then just rubbing the swab on the surface of the item, and you're attempting to collect any, in those examples I gave, any kind of skin cells or DNA that could have been transferred to the surface of those objects. So you do the wet swab, and then studies have shown that if you follow that up with a dry swab, that you can actually collect more DNA, because sometimes after the wet swab, there's still some DNA left behind that can be collected on that dry swab. And then both of those swabs are then tested in the DNA process.
And now when you're trying to collect DNA, you're actually collecting different types of substances from the body, correct?
Sure, yes.
And what kind of things would that be collecting? So can you go through examples?
Well, there's things like body fluids, right? So many times at crime scenes, you might have blood present. If it's a sexual assault case, we might be looking for semen or vaginal fluid or saliva. So those are all things that have DNA and typically body fluids have a larger amount of DNA than what we call touch or transfer DNA. Because what we've found is that, your DNA is present in all nucleated cells, so it's also possible to transfer DNA just from touching an object. And so when we do that swabbing method, again, we're trying to pick up either skin cells or DNA that has been inadvertently transferred to your hands. If you have a habit of maybe wiping your eyes or chewing on your fingernails or things like that, you can pick up even more DNA on your hands that can be transferred when you touch something.
So it can be saliva, it can be sweat, it can be skin, it can be anything.
Sure, yes.
And then what are some of the limitations from this traditional method?
So, a cotton swab is very useful in lots of different situations. They're cheap and easily accessible and, easy to use. But there's a limit to how much you can collect from on this cotton swab, right? Especially if you have something that is maybe a larger object. Maybe you have a clothing item from a victim that has been removed by the perpetrator, but you don't know exactly where that perpetrator touched the clothing item. With a swab, you could maybe pick certain areas. You could say, okay, I'm going to do the shoulder area, or maybe the back, the top quadrant of the back. But there's a limit to how much you can actually collect on that swab. So that's one limitation. If you have rough objects like brick or rocks or something like that, that swab really gets torn up, gets shredded, when you're trying to collect over a rough object. So those are some of the limitations. And not all of the DNA that's collected on the swab is released during the extraction process. The extraction process is not 100% efficient. It's not going to be able to get all of the DNA that was collected on that swab into solution. So those are some of the limitations.
Now, you said another way of doing it is through an MVAC.
Yes.
And can you basically describe, and I'll put up another diagram up here, what the MVAC does?
Sure. So the easiest way to get, kind of a visualization in your head about the MVAC is to think about when you get your carpets steam cleaned, right? You have a liquid solution that is being sprayed down onto the carpets while there's a vacuum being applied, and it's picking up all that dirt and gunk in the carpets, right? This is basically that, but miniaturized. The sampling head that we use is only maybe about the size of a 50-cent piece, maybe more like a quarter between those two. And it's single use, so it is a sterile, it's a sterile sampling head, only used one time, so as to avoid contamination. And we're not using steam, but we are using a sterile buffer that is being sprayed down onto the surface of the item. When that happens, it's basically loosening any of the cellular material that might be present, and a vacuum is being applied at the same time. So anything that is loosened from that item of evidence is then collected. It's sucked back up into the tubing, and it's collected in a sterile bottle. That bottle of solution that contains your DNA that was on that object is then passed through a filter. It's a very fine filter such that the DNA molecules cannot pass through that filter. They are basically stuck to the top. And so that's what's tested. You cut out the filter, and that's sent out to the DNA lab for testing. And the analysis process or testing process is exactly the same as any other sample, whether it's a cutting from a piece of fabric or a swab head. It's just in a different format. It's a small piece of filter.
So listening you describe that, me being just a layperson, if there was something on this carpet I wanted to clean up, it'd be the difference between taking a broom and dustpan versus, like you say, a wet vacuum, someone coming in and steam cleaning a carpet.
That would be a good analogy because, the swab, you're kind of just getting the surface, whatever's on the surface, whereas the MVAC, again, it's spraying down into all those cracks and crevices and, creating this kind of little mini hurricane of liquid that is then collecting any material present, whether it's DNA or dirt or anything, it collects everything, and then passing that into a bottle. That's where the DNA is collected in that liquid solution.
So you said that the cotton swab technique that's relatively inexpensive, and the MVAC, I assume, is a lot more expensive to own and operate.
It is, and so this is not something that you would use on every single piece of evidence. It's time consuming. Each piece and part is sterile, and most of it is single use, so it gets fairly expensive. The MVAC is best for porous items, so things like clothing or fabrics, the trunk of a car or something like that, where it would be difficult to swab, the trunk of a car, to swab the entire thing. Yet you could MVAC the entire trunk of the vehicle or back seat of the vehicle fairly easily. It's also good for anything where there's, like I said, rocks or bricks, things like that. If you had a smooth surface or something relatively small, you might want to just swab it. But it's the porous items where we tend to see an increased yield of DNA with the MVAC as compared to swabbing.
It would also be helpful if something has a recession in it, so it's not flat. So like as in looking at this table, this is enlarged, if I have something that has that crack built into it, that MVAC is better at getting into those cracks.
Exactly. I mean, that's basically what it's designed for, yes.
And you brought an MVAC, it seems here, and you're here to demonstrate for the jury how it just kind of works.
Yes,
Would you like to do that now?
sure. Okay, so I left all these parts kind of out for you so I can show you that this is not, if this were evidence, I would have a lab coat, a hair pulled back, a mask, gloves, the whole deal. This is just for demonstration purposes, so I'll just go without gloves even though it feels funny to me. So I'm going to go ahead and show you how it is put together. All right. So the components, this is the buffer solution. It is sterile. So it comes in this container where we know that it's been sterilized. It has a lot number on here, so I can keep track of the lot number. And this is what is going to be sprayed down onto the item of evidence being tested. It hangs right here. Next component is the tubing. So this is where the buffer passes through, and but it's also used to provide a vacuum. And again, this is sterile as well. Until I touch it, contaminated. Okay. So, goes into the MVAC here. And then this part next to the liquid. Right. And then this is the sampling head. And again, this is sterile as well. We can check with that. It's got this little mark on here that allows me to know that it has been through the sterilization process. And these are single use, so I would only use, each one of these is used only one time on a particular item of evidence.
So if you're going to sample 10 items, you need 10 of those.
Yes. So here's what this consists of. This is the sampling head. So the liquid is actually going to come out of here as I press it down onto the item of evidence. And then it travels through this tube and into this collection bottle. So the other end of the hose, the one end of the hose in the MVAC, the other end of the hose into the sampling head. Okay. And then you're basically ready to go. So what brought in was basically a strap, right? So this is similar to some of the straps that I sampled for this case. Much cleaner, but basically the same thing. So turn power on, solution pressure turns on, and that there's like a balloon, basically a bladder behind this that pushes forward. So even when you have very limited amounts of liquid in here, there's still a lot of pressure for that water when it comes out, or the buffer when it comes out. Once this light goes out, that means I have enough pressure that I can turn on the vacuum. Okay, and so you can kind of hear it hissing there now. Tighten this up. And let me show you first. This little lever here turns the water buffer on and off. So I turn it on, and it starts spraying out. Right? So I'm not sure how well you can see that, but I could probably hit you guys from here, but I will not do that. So what you want to do is you place the MVAC sampling head on the item, and you turn the lever on. So it's kind of like you think of when you're at the dentist's office and they put that thing in your mouth to suck out the saliva and water. That's kind of the sound it makes, and that's telling you that it's suctioning, it's collecting. So
Generally, how many passes would you do per item?
I usually do two. I do one with the lever turned on where the liquid is coming out, and then I follow it up without the liquid coming out but still vacuuming, so that I make sure I collect everything. And what's happening is, see, now I have some liquid in here. So the liquid is being collected as I do this. So if you can see, it's a little bit difficult to tell, but you might be able to see a color difference where this is now wet, right? And I can do this entire item, right? If I had a swab, it's going to be more difficult to collect from a swab. To do this whole thing with one swab, it perhaps could be done, but would definitely be more difficult. And I don't know if by the end the swab's going to be dried out. Am I actually going to be collecting anything or not? I might actually be transferring some of the DNA I just collected from here. By the time I get to the other end, I might be transferring it almost back onto the item. There's just a limit to swabbing. But with this, again, I can collect from the entire item. And then I can collect if there's any spray, because this is a sterile surface, and in the lab I would have a sterile surface as well. We can collect. All right.
Now, one thing you made the comment of that this is very clean versus some of the other stuff you tested.
Yes.
If something's dirty, is that a limitation for the MVAC?
Not really, no. It will still collect everything. The only kind of limitation is that the next step when I filter it, if it's got a lot of mud or debris in it, it can clog that filter because it's a very fine filter. So the manufacturers have developed something called a pre-filter where the DNA will go through that filter, but some of the bigger chunks of mud or debris, things like that, would not go through. And so that helps so we don't clog the filter.
And if something's dirty, is that a limitation on the cotton swab?
Sure. I mean, the cotton swab's going to collect the dirt as well. It's just, if I wanted to collect a very discrete area, if I knew that somebody tied this up and touched just this area, I could probably swab that. But I don't know where this was touched. So I want to, if I had a case where I don't know where it was touched, I'm trying to get the person who handled it, their DNA, I can do that in a whole item. So that's one of the key benefits, I think, of that. Along, again, with the fact that studies have shown that it collects more DNA, anywhere from, some studies show about 20 times more DNA as compared to a swab. One study, validation study, showed 200 times more DNA than a cotton swab. So the last step is to collect the DNA in the filter. And again, this is single use. It's sterile as well. So what it looks like, you can see in there's a white kind of circular thing there. That's the filter, right? So any DNA that I collected is going to pass through this filter, or excuse me, the liquid's going to pass through the filter, and the DNA would remain on that white part. So I'll just show you that very briefly. So I would take my solution, which sometimes I'll fill up 2 or 3 bottles worth of liquid, and it can all be combined under the same filter. Okay. So this vacuum hose now connects to this. Turn the vacuum back on. The liquid
assume the vacuum is making sure the water comes through that filter
Yes.
as efficiently as it can.
Right, and so this, I mean, you probably can't see it from there, but even with that small amount that I just did, I can see some blue fibers on there. So it collected the fibers, and so if there's any DNA on there, it's going to be collecting that DNA as well. So then what I would do is I would cut that filter out with a scalpel and package it up and send it, either test it myself at my laboratory or send it out to a state or local laboratory. So I work with some agencies, their crime labs don't have an MVAC, but the lab will process MVAC filters. And so I'll send it to their state lab. Or some agencies want me to do the entire testing. And I can do either one.
And so is it common for you to do this with state or local agencies as a contract?
Yes, so I do a lot of whole case work. And most of that, all of that, frankly, so far has, one case was from defense, and then that was more a post-conviction case. And then the remainder of the times I've used MVAC, with the exception of this case, have been for law enforcement agencies, prosecution.
You done with the?
I am.
Okay. Is it fair to say that the MVAC is prominently used in, I guess, government agencies?
Yes. So there's a number of MVACs that are being used by crime laboratories, and then there's probably a larger percentage that are being used by law enforcement agencies at the scene of the crime, or in their facility, they will do the collection at the police station and then send the filters to their state or local lab if that lab does not have an MVAC.
Now, if I was the captain at an agency and I said, I want to buy one of these MVACs from my agency, and I do, I assume I would have to go through training, or my staff would either have to be trained and certified on how to use it. Would that be fair to say?
Yes, you would need training on how to set up the MVAC, how to use it, just as I had done. That was something that was provided to me by the manufacturer of the instrument.
But would there also be a concern of contamination?
Sure, yeah. Just like with any DNA testing, you have to be careful that you're handling everything properly, that you're not getting your own DNA on the item, because it's so good at collecting DNA. It just would take a very small amount of your own DNA. It would collect it, and you could have contamination, especially with the kits that are being used today, because they're very, very sensitive. So with the kits that are currently being used, you can get results from 19 or 20 cells worth of DNA. Then you combine that with a very sensitive collection method, you really increased your ability to obtain results. But yeah, you certainly have to be careful with avoiding contamination.
Simple question. You said you can get it from maybe 19 cells, you can get DNA. If there was a drop of blood, just a normal sized drop of blood on a table, how many cells would be approximately in just a drop of blood?
I know that there's probably 1,000 or more nanograms of DNA, and there's 152 cells per nanogram, so probably 152,000 cells probably in a drop of blood. I mean, it would depend on the size of the blood, etc.
So this is getting microscopic level of DNA sampling done on an item.
Right, and so, yeah, and that's typically what we see with touch DNA. It's typically a lower amount of DNA than a body fluid.
And so, getting to now this case, you were brought on as a consultant, obviously by me, to review the DNA evidence that was presented or developed by the government.
Correct, yes.
At some point, was there a suggestion made, maybe we should test some of the items?
Yes,
And were you involved in that process?
yes.
And what were some of the things that you were thinking about when you were making a decision on what items should be tested?
Well, looking at the evidence, I saw that some of the items had been tested, and some of those items, none of them yielded a DNA profile. Some of them did show really low levels of DNA. So my thought was, what, if we use a more sensitive collection technique, perhaps we can get enough for a profile. And so I wanted to focus on items where we thought the perpetrator would have had contact, would have applied pressure or friction. And so initially, we talked about like the electrical cords, the tie-down straps that were present that had been found on some of the victims, on some of their bodies.
The items that were used to wrap around, I think it was Joseph McStay, the blanket, and everything that was wrapped around that.
That was like holding it together, yes. Right.
And some of the other items you thought would be worthwhile testing.
I had also suggested possibly some of Summer McStay's clothing, especially the bra, since it had appeared to have been cut, based upon the photos that I had seen, that it was in two separate pieces, and the underwear, because I felt that perhaps those items would have been handled by the perpetrator.
Since they were taken off her body and found near her head and not on the actual body.
Objection, the state's testimony or explanation.
Guys, take it off her body.
They were not on her body when they were located.
Right, that was my understanding.
And so you requested items to be sent to you from the San Bernardino County Crime Lab, correct?
Yes,
And those items were sent,
yes,
and they were received by some kind of courier to your laboratory?
yes. I received them, yes.
And the items you tested, you used an MBAC as you demonstrated here on those items,
I did, yes.
but you followed more of a sterile environment.
Yes, very much so.
So I'm going to go through some of the items that you tested that yielded results.
Okay,
One of the items that you asked to be tested was previously marked as item E11, a white cord.
yeah, is it okay if I refer to my report while we go through these?
Would that help refresh your memory of everything?
It would, yes.
Yes. And if you need to do that, just let us know you need to review the report before you answer.
Okay.
Okay, so I have before you Exhibit 994. You can see it on the screen to your left,
Okay.
so you don't hurt your,
Oh, yes.
your neck.
Thank you.
Do you recognize this photograph?
I do, yes.
And how do you recognize it?
I took the photo, those are my initials and the date. And the
So this-
item number.
And this was an item you received from the crime lab?
It was.
And it was received from you under barcode number 137001773.
That is the barcode that the crime lab used in their reports. I can't say that that barcode was on that particular item of evidence.
When you received it, it was in an envelope, correct? Like
It
this,
was,
right? Let
I
me-
don't know if it was in a box or an envelope right now.
Okay, let me back up. When you received the items, describe what you received.
So I received 3 large boxes and 1 smaller FedEx box.
And so when you received those boxes, what did you do?
I opened the boxes individually to begin processing the items of evidence.
Okay. And were the items marked so you can identify which items were which?
Actually, not all of them, no. It took some deductive work to figure out what all was being submitted. Some were marked on the exterior of the box. Some I had to look back at the analyst's notes and photographs to ensure that I was working with the same item of evidence.
So they sent you things that were just kind of sometimes thrown in a box or thrown in a bag, not marked?
Objection.
Yes. Oh, sorry.
Overruled.
So they were able to figure out that this item was E11.
Yes.
And did you review the bench notes and the report of Don Jones and the work that was done by the crime lab on the items that you had sent to you?
Yes,
And based on the testimony of Don Jones, he said that he actually analyzed this item
yes,
with the traditional method.
with a swab, yes.
And he said there was no DNA found on this item.
Right, no DNA profile was developed, yes.
And then you processed this item?
I did.
And how did you process it?
I used an MVAC, just like I demonstrated there. I processed the entire length of the item,
And one side or both sides?
both sides.
And also up near the head here, where the male part of the extension cord or the white cord would be.
Yes,
And after you process that, you process the filter then?
so then I allowed the filter to dry down. I cut the filter out and packaged it as an individual item that was then sent to a different lab for DNA testing.
And what item number did you give the filter?
E11A.
And then the second item that you tested, you recognize as shown exhibit 995, correct?
Yes,
When I say second, I'm not saying this was the order. Would that be fair to say?
the order of what?
The order you test them. When I say second, doesn't mean this was done
Oh.
next.
Correct, so this was, yes.
And so this is the next item, and this photograph 995, what does this show?
This is a pair of underwear.
And is this the picture you took?
It is,
And that represents item A2 that was tracked by the Sheriff's Department?
that's correct.
And in reviewing the testimony of Don Jones and his report, did he try to analyze these items?
No, I don't believe he did.
And did you process this item?
I did.
And how did you do that?
I processed the entire interior and exterior crotch area. I processed the waistband area. Basically any area that I could with this item. It was very thin and kind of fragile. The material was very thin, but I did process the entire item.
And at some point, did you let the filter dry and cut that out?
I did.
And did you label that item?
I did. That I labeled as item A2A.
Go to the next item that was processed, which is exhibit 996. You recognize that item?
Yes, I do.
And what's that exhibit show?
That is a partial black bra. That was the left cup and strap of the black bra.
And did you take that photograph?
I did.
And did you process this item?
I did.
Well, let me ask you, did you review Don Jones' testimony and note that he processed this item?
I believe he did. I probably want to refer to the notes to be certain about that.
And are you referring to the notes of your notes, or?
No, Mr. Jones's report. Would that be okay?
Yes. Would that help refresh your memory?
It would, yes.
Do you have those with you?
I do.
Okay, perfect. I was trying to find it.
Oh, okay.
I'll stop.
Okay, so that item actually was not swabbed for touch DNA. It was evaluated, so that meant he looked at it to determine if he thought, you know, he wanted to swab that particular item. But it was not swabbed.
And did you process the item?
I did, yes.
And how did you process it?
In the same manner. With the MVAC, I did both interior, I basically did the entire item, both interior, exterior, the strap, the shoulder strap, and the back strap, and the clasp.
And the cup as well?
Yes,
And then that item, which is A3, correct? That was labeled
Yes.
by the Sheriff's Department?
yes.
After you're done, did you let the filter dry?
I did.
And did you cut that out and label that?
Yes, I did.
And how did you label that filter?
That was A3A.
On to the next exhibit. You recognize exhibit 997?
I do.
And what is that?
That is item E15.
Was that a picture you took of that item?
Yes.
And E15 is another electrical cord that was removed from Joseph McStay.
That's correct.
And you're reviewing the testimony and the reports of Don Jones. Did he analyze or process this item?
Yes.
And what did he put in his report?
The item was processed and that the total, basically the amount of DNA that was detected was, excuse me. He took that sample through the quantitation stage of analysis to determine if DNA was present. But no DNA was detected, so he did not move that forward through the analysis process.
So he said there was no DNA found,
Correct.
but there was a quantitative analysis in his report. What does that represent?
Right, so in the bench notes, which is where the quantitation results are contained, there was an extremely low level of potential DNA detected when the quantitation is done, we look at how much human DNA is present and how much of that is from a male. The human DNA did show undetected. The male DNA target showed an extremely low level of DNA present.
So according to the test he did, there was still some male linked DNA on that item on his test.
There was some detected. It was above the undetected level, but below the standards of the test procedure. So yes, there was something detected at a very low level.
And did you process this item?
I did.
And how did you do that?
I back the entire item, loosened the knots so that I could access underneath that knotted area to see if there was any DNA underneath that particular area.
And did you let the filter dry in this manner?
Yes,
And did you cut it out and label it?
I did.
And how did you label it?
This was E15A.
Exhibit 998. Do you recognize that exhibit?
I do.
And what does that exhibit show?
That is a red strap.
And does it have an item number associated with it?
Yes, A25.
And did you review the testimony and the notes of Don Jones, whether or not he processed this item for test DNA?
I did. He did not process this item.
And did you process it?
I did.
And how did you process it?
With the MVAC, so I collected from both sides of the strap, the entire length of the strap, went onto one MVAC filter.
And then that filter was dry,
It was.
and it was labeled.
Yes.
And what was it labeled?
A25A.
Did you find anything else while you were MVACing this item?
Yes, there was an apparent hair that, after I had spread the item out and to examine it and start processing it, I did see a hair that was on the bench paper that had apparently fallen off of the item. So I did collect that and put it in a separate package.
And so you didn't do anything with the hair other than reattach it and send it back to the crime lab, correct?
Yeah, so I put it in a separate sealed manila envelope, attached it to the outside of the package, and noted in the report that I had collected this apparent hair.
And did we get to if you labeled the filter?
I believe so, but it was A25A.
And last exhibit, exhibit 999. Do you recognize this one?
I do.
And what is exhibit 999?
So that's A26, which is the other half of the bra. It's the right cup of the bra.
And this is the photograph you took?
Yes.
And you reviewed Don Jones's notes and testimony if he processed this item?
Yes, he did process this item. And he was not able to get a DNA profile from this item.
He said there's no DNA found?
That's what his report says, yes.
Did you check the bench notes for a quantitative analysis on this as well?
Yes.
And was there one?
There was a very low level of human DNA detected. It was, again, below the lowest standard that we use in the quantitation kit, but it was indicating a low level of DNA present.
And it was actually a higher level than the one that was on the cord though, correct?
That's true, yes.
So did you process this item?
I did.
And how did you process this item?
For this particular item, I processed the front of the bra and the strap and the clasp. So both sides of the strap and the clasp, and then the front of the bra.
And why did you only process the front of this one?
This one was in better, in my opinion, kind of better condition. And so I'm looking for DNA foreign to the person who was wearing this bra. So I thought, well, I would focus mostly on the outer wear and see if we could pick up any DNA from someone who had come into contact or touched the bra.
And so then, have the filter dried on this item?
I did.
And did you cut it out and package it?
I did.
And did you label it?
I did.
How did you label it?
As A26A.
Now, all the items that you received at the crime lab, from the crime lab, now there's other ones that you sampled, correct?
There were.
We won't go through those because we're going to skip that. The items that you received and processed, did you repackage them?
I did.
And did you send them back to the crime lab?
I did.
And then the filters that you cut out and labeled, what did you do with those?
Those filters were sent to a private DNA lab, Bodie Technology.
Yeah.
Actually, it was Bodie Cellmark at that time.
And are you the one, were you involved in any way in selecting what lab would do the analysis?
I believe so. Yeah, because I typically do send samples to Bodie just because I used to work there a long time ago, but I also know, their procedures, and I had a lot of trust in the capabilities of that laboratory. It doesn't mean that other laboratories are not capable and good labs. It's just one I was comfortable working with.
Is it fair to say it's probably the largest private lab in the country?
Yes,
Now, you described earlier that you have your own certified lab.
I do now, yes.
So did you at the time you did this testing?
I did not, no.
And so you couldn't do this yourself because you just didn't have a certified lab.
Right, exactly. At that time, yeah, we had not, my report, I wrote in March of 2018. I was working at the other lab, but we were not yet accredited. So I did not want to do the DNA testing there because we were not an accredited facility at that time. It wasn't until August of 2018 we received our accreditation.
And when you sent the items to Bodie, how did you send them?
Via FedEx.
And do you have the tracking number of the date you sent it?
Yes, I do.
And what was the tracking number?
I'd have to refer to my report.
Would that help refresh your memory?
Yes,
Do you have that report with you?
I do.
You can please review and let me know when you're done.
Have to memorize it.
If you could just read it off, because it's the number.
If that would be okay, yeah. 7717-6616-2370. And I sent those on March 9th, 2018.
Now, part of Don Jones's testimony was that he said human DNA would never survive in the ground after 4 years. Did you hear that part of the testimony?
Yes, I did.
Do you agree with that statement?
Well, I've done a lot of research lately on trying to determine, what do we know about the persistence of DNA after it's been buried. Unfortunately, there are very limited studies. The studies that I've come across are dealing with bodies and bodies placed in the ground and how long does DNA last from tissues on the body or from soil. So some of the studies, the bodies have decomposed in the soil or on top of the soil, and then they've tested the soil to see, can we get DNA from the soil? We can for some time period. One of those studies showed, I believe it was 172 days later, or almost 200 days later, they're still getting DNA from the soil, but those don't really relate to what we're talking about here, which would be, the trace or touch DNA on clothing that's been buried. The closest that I found that could kind of relate was someone had done a study in 2009 where they took...
an objection as to the study.
And you can give us your opinions based upon all of your studies and research,
Okay.
but you can't just say, well, here's a study and here's what this study says.
Okay.
So it's the same as study.
So there was a study that you found that kind of addressed the issue, basically?
Same objection.
Overruled, you can answer yes or no.
It was the closest that I could find. It was by an author named Borja.
Objection,
Borja.
move to strike.
She can identify the study, Your Honor.
Overruled, answer remains.
The author was Borgas. This particular author had dried down blood stains on different types of fabric and then buried
Objection.
Overruled.
and then buried them in different types of soil. Marshy soil, clay soil, or in sandy soil. And he then dug that up at 15 days, 30 days, and 90 days. He was still able to get results. It was degraded, meaning broken down, but still able to get results at 30 days and 90 days.
Now,
That's the longest he did. So whether that relates to years later, frankly, I don't know. Those studies don't exist.
would it be fair to say to offer an opinion definitively without some kind of study for it, would you assume that'd be speculative?
Yes.
Now, do you have any experience with cases a body was buried underground for an extended period of time?
Well, I have experience with testing bone samples. I do that have been buried. The only case that would be similar to this would be a case where clothing was buried. It was buried from, I believe, 1983 to 1990 when the clothing was unearthed underneath a person's home. I M-backed those items of clothing. The results were very low level amounts of DNA, so low that the lab chose not to go forward with testing. However, there was a hair that was located on the clothing item, and after burial for at least 7 years, that the lab was able to get a partial DNA profile from that hair. So that's the closest I could, in thinking about my past experience, that's the closest that I could relate to you.
Now, two of the items that were processed by Don Jones had DNA under his processing that survived, right?
So when we do the quantitation, it is an estimation of the amount of DNA. It is a sensitive method. It was detecting a very low amount. It was so low that he was not able to get a profile, but there was, on two of those items, some DNA detected.
And now, you made a comment or referred to a term of DNA being degraded. Can you explain to the jury what that term would mean?
Sure. DNA breaks down over time. So, you know, your DNA molecules are very long stretches of nucleotides, of molecules connected together. And those can break down over time. So then you have shorter stretches or pieces of DNA. The type of testing we do is actually very effective even on degraded or broken down DNA because we're not copying the entire DNA molecule. We've got these certain specific areas that we're copying. The largest one of those areas is only about 400 base pairs in length, and the smallest is maybe 120 base pairs. So it's these little chunks of DNA. But sometimes the DNA is so old and so broken down that even those 400 base pair lengths of DNA, there's a cut in it, there's a break. And so the copying process cannot proceed, and you might not get results for those larger loci or those larger areas of DNA, but you might still be able to get results at the smaller loci. So when we have a degraded profile, you're more likely to see results on basically if you're looking at the data on the left-hand side of the page, and on the right-hand side, where the larger loci are, you might have very low results or no results. We call that a ski slope effect, where you have DNA here on the left side, and then it kind of drops off to nothing.
Now, when an analyst says, you know, there could be no DNA that survives in the ground that long, those are really assumptions. Would that be fair to say?
I would say in this case, just again, because, I mean, that was a question I had too, right? Are we even going to be able to get anything? And there wasn't any research that told me one way or the other.
Were there other assumptions about DNA, let's say touch DNA, that has been proven wrong over time?
Yes. So initially, people knew that you could get DNA. Scientists, we knew you could get DNA from blood and semen and saliva and bones and hair, but touch DNA, just DNA from somebody touching something, no one, you know, we didn't think that you could get a profile from something like that. And it wasn't until 1997 when the first researcher said, well, let's try it out and see. And they were able to get DNA results from touch DNA.
So that assumption was proven wrong.
Yes, yes.
Are there other examples of DNA assumptions that were made that were proven wrong?
Yeah, I mean, I'd say along that same lines, once we knew that you could get DNA from touch DNA, in that same original paper, they said, you know what, we're seeing other people's DNA. We're seeing what we think is secondary transfer. And a lot of people in the DNA community said, no, that's not possible. It's not sensitive enough. There's not enough DNA. And for many years, people did not think that secondary transfer, so an indirect transfer of DNA, was possible. But now, every year, there are more and more research articles that come out that show that, yes, secondary transfer is very possible, and even further transfer. So that's another thing where we kind of assumed that probably not going to happen. And there are other examples. Fire cartridge casings.
Before you go on,
Sure.
the development of the ability to see secondary transfer, does the development of the technology of DNA help with that?
It definitely does.
And
Yes.
could you explain how those advances have made a difference?
Yes. So as the technology gets more and more sensitive, so those amplification kits we use are more sensitive today than ever before. They're better at overcoming degraded DNA or something we call inhibitors, which is something that slows down the copying process. Today's kits are much better at overcoming those sorts of things. They're more sensitive. The instrumentation that is used to detect the DNA is more sensitive as well. And so when you have that increased sensitivity, then it makes sense you're going to see more instances of secondary transfer or DNA. You know, if two people shake hands, they could be transferring each other's DNA. You know, I could shake your hand, and now your DNA is on my hand, and then I touch this water container. I might transfer both my DNA and some of your DNA to that item. You never touched that, but your DNA might be there. That's a secondary transfer.
Okay, now you said there's other items or other DNA assumptions that were found to be possibly wrong, or when you start talking about fired cartridge casing?
Right. So for many years, it was thought in the field that you can't get DNA from a fired cartridge casing of, the shell or a cartridge casing after that's been ejected from a gun, because the heat that was produced would be so high it would destroy the DNA. And in, I think it was 2004, there was a very small scale study that was reported in a newsletter, basically. And they said, yeah, we really didn't get any results. And I think a lot of people kind of said, oh, okay, yeah, you're not going to get results from that. And it wasn't until additional studies were conducted with maybe a more sensitive kit that we found that, yeah, you can get DNA, not on every sample, but about 35% of cartridge casings, you can get a DNA profile. Things submerged in water. Just recently, we found that, you would think touch DNA or DNA that's just been transferred from skin cells, if you take an article of clothing and put it in water, it would kind of make sense, well, that is going to kind of dissolve the DNA or make it float away. What we found recently is that in certain conditions, which would be colder water, slow-moving water, that DNA can last between 10 and 14 days. So I guess the point is, until the studies are done, we can't just assume that there would be no DNA present.
Now, going on to transfer DNA, there's been a number of studies about transfer of touch DNA. Would that be fair to say?
Yes.
Are you familiar with the Lucas Anderson case study that worked on it?
Yes. So that's actually a real life example of secondary transfer, because sometimes the argument is, well,
Discovery and non-responsive.
The objections stayed on the same basis again. She can offer her own opinions, but can't simply relate individual studies.
One of the studies I proposed to Don Jones was one that was published in the Forensic Science International Genetics in 2014, discussing the persistence of DNA deposited by the original user on objects after subsequent use by a secondary person. It was written by numerous people, including a Von Oorschot, O-O-R-S-C-H-O-T. You know which study I'm referring to?
I do, yes.
And was that study helpful for your analysis of touch DNA as it relates to this case?
Yes.
And whether, basically, what did that study deal with?
In that particular study, they were trying to determine how quickly would a second user of a particular item, how quickly would their DNA replace the first user. So they looked at porous items, it was like a bracelet, and then a non-porous item, which was a pen. So they had a first user handle just a plain, blue ballpoint pen, handled it for a total, I believe, of 210 seconds by rubbing it between their hands on 3 different occasions and taking the lid off and putting it back on. Then they had a second user use that pen for defined periods of time. And then they would swab the pen and see, do we still see the first user, or has that second user kind of taken over as the major contributor?
Based on that study, could they develop like a time frame when the secondary user's DNA starts overcoming the first user?
Yes.
And was that part of the study that became informative for you about what to expect from a touch transfer DNA circumstance with somebody handling an item?
Yes.
And so, what are the time frames that they really noted as important?
Well, even between 1 and 5 minutes, they saw it was almost 40% or more, almost 50% of the DNA was from the second user. But it was at 30 minutes that that second user became the major contributor. And at 90 minutes, the first user was only about 15% of the profile. So 85% of the DNA on that pen at 90 minutes was the second user.
So what you're saying is the longer somebody held on or used an item, the more prevalent their DNA became on an item when you like analyzed it and see whose DNA was present. Would that be fair to say?
Right, that was the general trend, yes.
And you're talking about 50%, 85%. You're referring to mixtures?
I am, yes.
And can you explain what you mean by that? I know Don Jones testified about that briefly, but can you explain that?
Sure. So a mixture is simply DNA from more than one person. It might be a mixture of just two people. You might have two, 3 people, 4 people. It's typically we can tell you the minimum number of contributors to a sample, but sometimes it's unclear exactly how many contributors are on a sample. So in the Penn study that I talked about, they were discussing major and minor contributors, and they did see some other foreign DNA not matching either participant that was just inadvertently transferred, probably from someone's hands that they had picked up somewhere else.
So for part of the study that really dealt with this issue was a non-porous hard surface. Would that be consistent with the steering wheel?
I mean, it would depend on the type of steering wheel. If there wasn't a cover, a cloth cover or something like that on the steering wheel, then yes, I think it could be consistent with the steering wheel.
So if a vehicle is driven a distance and it's approximated about 90 minutes to get from one location to another, if that person, whoever's driving that vehicle from one location to another, without gloves on, barehanded so they can leave sweat or whatever, touch the skin, any kind of touch DNA, would you expect to see that person's DNA in that mixture if there was any mixture pulled from that steering wheel?
I think it would be possible to see that person. Based upon the study, I would say it would be more likely to be the more likely to be the major contributor if that person had driven, for 90 miles or 90 minutes.
90 minutes. And you're familiar with the testimony. Don Jones says that Mr. Merritt was a minor contributor from the steering wheel, correct?
Right.
With approximately 21% contributing to the mixture.
Yes, correct.
Would that be consistent or inconsistent with your expectations based on the studies and if it was a 90-minute drive?
That would be inconsistent with at least that particular study. If that person had driven...
There's been no discovery on this, but she's relying on this particular study. So there's a couple different studies out there. I'm trying to figure out which one.
We've only talked about the one, the 2014.
Not the 2016.
No, just the 2014. We haven't mentioned the other two.
So I would say, although the 2014 study that we're discussing did not involve steering wheels, it involved a hard, non-porous surface. If their results hold true for steering wheels, also a hard, non-porous substrate, then if someone drove a car for 90 minutes, the expectation would be that they would be more of the major contributor.
Before we go into another one, the court want to take the lunch recess now?
Sure, it's about noon. We'll go ahead and take our noon recess at this time until 1:30 this afternoon. Just a reminder, we are going to recess today just before 3 o'clock for one of our jurors. So we'll see everyone back at 1:30. And again, do not form or express any opinions about the case. Do not discuss the case. See everyone back at 1:30.